Welcome to our webinar, Catalyst for a Cure 3, Vision Restoration Research Update. My name is Tom Bruner, and I am the President and CEO of Glaucoma Research Foundation. Catalyst for a Cure is Glaucoma Research Foundation's leading research program. This unique collaborative approach to research has led to groundbreaking discoveries for glaucoma and the medical world. In 2002, we launched the first Catalyst for a Cure team. They were among the first to show glaucoma is a neurodegenerative disease, not an eye pressure disease. This was followed by the Biomarker Initiative, where they identified novel indicators of glaucoma, which help clinicians detect, measure, and treat glaucoma with unique precision. In 2019, we launched the third Catalyst for a Cure, to cure glaucoma and restore vision. The 4 investigators have shown extremely promising early results that may lead the way for new genetic, neuroprotective, and cell replacement therapies. In 2022, a fourth initiative, the MELSA-M and Frank Theodor Barr Catalyst for a Cure to Prevent and Cure Neurodegeneration, was launched. These scientists are investigating several neurodegenerative diseases, including glaucoma, seeking common mechanisms that could lead to cures for glaucoma, Alzheimer's, and other neurodegenerative diseases. Today, we will learn more from 2 of our accomplished Catalyst for a Cure vision restoration principal investigators, Drs. Ana Latorre and Dr. Derek Welsby. Dr. Latorre attended the University of Barcelona in Spain, where she received a master's in cell biology and a doctorate in neurobiology. Today, she is a professor in the Department of Cell Biology and Human Anatomy at the School of Medicine at the University of California, Davis. Dr. Derek Welsby attended the University of California, Los Angeles, where he received his medical degree and PhD in molecular biology. Derek is an associate professor of ophthalmology at the University of California, San Diego Shiley Eye Institute. Both are truly amazing scientists, and it is an honor to have them with us to share their Catalyst for a Cure research. Dr. Welsby will speak first, followed by Dr. Latorre. After their presentations, we'll discuss their research and respond to some of the submitted questions. Derek, the floor is yours.
Thank you so much, and also to the Glaucoma Research Foundation for all of the support over the years, and of course to my 3 collaborators, Ana Latorre, Shinduan, and Young Hu, have just been an amazing team to work with. So I wanted to give a little background first to help understand the topic we're going to discuss today. This is an eyeball that you're seeing right here. And of course, everyone knows that. And it focuses light onto the back of the eye, onto a tissue called the retina. If we use the analogy of a camera, the retina would be like the film in the back of the camera. And it's composed of many types of cells, one of which is an optic nerve cell, also called a retinal ganglion cell. And each of these optic nerve cells has a very long fiber called an axon that connects that point on the retina to a point in the brain. Now, I'm showing you one cell, but in reality, there's about 1 million of these cells lining the retina, with each one representing essentially a pixel of your vision. So the way this works, light is focused onto the retina, visual information is processed and transmitted down that fiber to the optic nerve, to the brain, where we then experience vision. So what happens in glaucoma? This is the retina as your ophthalmologist sees it. Now I'm going to overlay a cartoon of those optic nerve cells and what they would look like. And again, I'm showing you 12. I'm not showing you all 1 million just to make things more clear. In glaucoma, those nerve fibers or axons get damaged at a structure called the optic disc. The fibers degenerate, and ultimately the cell that has just become disconnected from the brain figures, it's not so useful anymore. So it actively commits suicide. And this process continues until you lose more and more of these optic nerve cells, moving closer and closer to the center of the retina. The disease is of the optic disc, combined with the loss of optic nerve fibers, leads to this characteristic shape of the optic disc we call cupping. And because these are nerve cells, their loss is permanent. They don't grow back. And for some patients, they eventually lose all of these optic nerve cells. Now I'm going to show you that same animation, but synced up to everyone's favorite test, the visual field. A patient stares straight ahead. We shine lights in various positions to test which optic nerve cells can still be activated by light. The field here is totally normal. The black spot is normal. That's the physiologic blind spot. And remember that the optics of the eye inverts the image. So if there's problems on the bottom of the retina, it manifests with something in the patient's upper visual field. So early on, even though you have damage and even some degeneration, we don't detect that on our visual field because you have more optic nerve cells than you need. But at some point, you lose enough of these cells that parts of the retina are simply disconnected from the brain, and you develop visual field defects. The first cells that die aren't in the center of the retina, which is why patients lose peripheral vision first. But more and more cells die, more and more of the retina becomes disconnected, moving closer and closer to that center of the retina that we use for reading and recognizing faces, until eventually central vision is impaired. And for some patients, sadly, they lose even the ability to sense the brightest of lights. So the CFC team, sponsored by the Glaucoma Research Foundation, has been very focused on new therapeutic options for these patients. There are really three challenges we're trying to tackle. The first is to rescue cells from death. It's what we call neuroprotection. But for those patients who have already lost their optic nerve cells, that's not going to work. So we need a way of replacing the lost cells. And finally, we need a way of getting those replaced cells to form the correct connections and grow back to the brain. As you can imagine, it's easiest to save the ones you've got, and it's hardest to put back the new ones. So today I'll be talking about updates on that first challenge, rescuing cells from death. And then Dr. Ana Latorre will give you an update on the second challenge, replacing lost cells. So how has the team gone about figuring out how to protect optic nerve cells from glaucoma? Remember this little diagram that I showed? It's actually a little misleading. It shows all those optic nerve cells in the same way. They're the same shape, the same size, same color. In actuality, it'd be more accurate to show a diagram like this. There are actually many different subtypes of optic nerve cells, and they do different things. And so then the question is, do all these subtypes respond to glaucoma in the same way? So the GRF team, led by Dr. Shin Dewan at UC San Francisco, figured out a way to watch these optic nerve cells die in real time using a rodent model of glaucoma. This is before each one of the little white dots is an optic nerve cell. And this is shortly after the animal experienced elevated eye pressure. You can see that some cells highlighted with blue circles die off, and other cells highlighted with the yellow circles don't die. I'll flip back and forth so you can see. So, of course, that begs the question, what's different about those cells that are highlighted in blue and highlighted in yellow? So using some genetic tools, we can keep track of the various cell types. For instance, some cell types, like subtype number 4 here, were very susceptible to glaucoma. This is the number of cells before glaucoma was induced, and this is afterwards. And you can see that a lot have died off. In contrast, another subtype, like subtype number 1, before and after glaucoma look pretty similar. So really the question is, what is different about those that live versus those that die? And the team identified a unique protein called osteopontin that was present in the resistant ones, but not in some of the susceptible ones. Now, if the protein is the cause of the resistance, we should be able to make 2 predictions. First, if we subtract that protein from the resistant cells, they should now become sensitive. Conversely, if we add the protein to the sensitive ones, they should now become resistant to glaucoma. So we used CRISPRs, or molecular scissors, to remove the gene for this protein in the resistant cells. And then Shin's team repeated the glaucoma experiment. If the protein was present, the cells were still resistant, meaning before and after glaucoma, roughly the same number of cells as shown here. But if the protein was removed with those molecular scissors, the CRISPR approach, then the cells died off with glaucoma. They became sensitive to glaucoma, as shown here. So then what about the reciprocal experiment? We added back the protein using a gene therapy approach. You take the gene of interest, put it inside a safe virus. We call this a gene therapy vector, and then use that virus to infect optic nerve cells so that they express this gene of interest. And of course, the gene that we're going to express is osteopontin. If you look at the sensitive optic nerve cells, normally they don't have that protein, you can see that glaucoma still kills them. However, in the cells that express this resistance protein, many fewer of them die off. And this suggests that osteopontin is a potential neuroprotective gene therapy that can render optic nerve cells resistant to glaucoma. Now I want to tell you about a different story from the CFC, led by Dr. Young Hu at Stanford. There are these families, very rare families, that have an extremely strong genetic form of glaucoma. And it's been known for a long time, they have a mutation in a gene called optineurin. And it causes glaucoma despite relatively low eye pressures. So the team wanted to understand the reason for this link between optineurin and getting glaucoma at low eye pressures. Moreover, the team wanted to ask whether it could teach us something about why glaucoma is happening at low eye pressures, even in more common or what we call sporadic forms of the disease. So the team, led by Dr. Young Hu, made a mutation in the optineurin gene in mouse. They showed each of these red dots is an optic nerve cell in a mouse retina, and they showed that the optic nerve cells die over time, just like in human glaucoma, and that the optic nerve has fewer and fewer axons, just like human glaucoma. So what is this optineurin thing? It's a protein that associates with mitochondria. These are the bean-shaped organelles, which is just a fancy word for part of a cell, that act as the cellular powerhouse. Young's lab labeled mitochondria in red and then looked at the optic nerve of these mice. In normal mice, you see tons of mitochondria down the optic nerve, powering that axon or nerve fiber. But in mice with the optineurin mutation, you saw reduced amounts of mitochondria in the optic nerve. So why would that be? Well, mitochondria are transported to the fibers of the optic nerve cells using things called microtubules and molecular motors. If you want an analogy, think of microtubules as a highway system and the molecular motors as little semi-trucks, and then the mitochondria would be the cargo. Shown here is an animation showing how these molecular motors walk down those microtubules to move their cargo. Here, the cargo happens to be something called a vesicle, but if you imagine that to be a mitochondria, it would be essentially the same picture. So Young's team showed that optineurin is a key part of that tethering machinery, a key part of that little semi-truck that transports the mitochondria down the microtubule. When it's mutated, the mitochondria don't attach as well to the microtubules, so they don't get transported down the nerve fiber. And since the mitochondria are the powerhouses, amongst other things, that could lead to impaired energy production of the optic nerve, and that could cause the nerve fibers to degenerate. The next thing they did was to test whether they could use gene therapy to put back the components of that tethering machinery and treat the disease. Indeed, expression of the components of that machinery did improve the number of surviving optic nerve cells in this lower IOP model or eye pressure model of glaucoma. Also worked for axons and also partially rescued vision, suggesting that in animals with impaired optineurin function, you could reconstitute optineurin with gene therapy and treat the disease. Of course, maybe that's not totally surprising, right? If you create the disease with broken optineurin, I suppose it makes sense that putting back the correct version could help. So Young's team went one step further and asked, what happens in more typical glaucoma situations? So this is a different mouse model of glaucoma. It has nothing to do with optineurin. In this one, the eye pressure is elevated. And again, Young's team looked at the number of mitochondria in the optic nerve. And even though there's no genetic defect in optineurin, you can still see there are fewer mitochondria in the optic nerve compared to before glaucoma. So it almost seems like mitochondrial transport to the optic nerve and a defect in that process might be a general theme of glaucoma. So what happens if you use AAV gene therapy to increase the number of molecular motors, to increase the number of little semi-trucks? What you can see is that it helped to restore mitochondria delivery to the optic nerve. And it wasn't just that it helped the mitochondria. If you actually look at the number of surviving optic nerve cells, you see that the model, when you raise the eye pressure, reduces the number of optic nerve cells. But if you do the model, meaning you raise the eye pressure and you increase the number of molecular motors, you rescue the effect. So in conclusion, for this part of the talk, the team led by Dr. Shin Dewan used the differential susceptibility of optic nerve cells to identify candidate neuroprotective genes. This led to the identification and validation of osteopontin as a potential neuroprotective gene therapy. The team, led by Dr. Young Hu, used the genetics of glaucoma to identify a candidate neuroprotective gene, optineurin, and also its neuroprotective role as a glaucoma therapy. Now I'd like to hand this over to someone who I have had the very good fortune of collaborating with for the last 5 years, who is a wonderful collaborator, Dr. Anna Latore.
So thank you so much for the first introduction. It's been an honor to be part of this team. And as Dr. Welsby explained, we have been looking at different approaches to treat glaucoma. So as Dr. Welsby introduced very well, the hallmark of glaucoma is the degeneration of first these fibers, but also these cells called retinal ganglion cells. And as he also introduced, at early stages of the disease, when these cells are starting to get sick, the best approach probably is neuroprotection, that trying to preserve what you already have. But as he mentioned, at the late stages of the disease, the cells are no longer there. So at these stages of the disease, neuroprotection strategies are not going to work. And instead, we're going to have to use something different called cell replacement, which means introducing new retinal ganglion cells into that diseased retina. And this is, of course, very challenging. And I'm going to tell you how we are tackling the challenges one by one to try to develop these technologies. And so all this work has been done with the wonderful team of the Catalyst for the Cure 3. And so what are the challenges? To replace these cells, first, we need a source of cells. We need to get the cells that we inject into the eye to survive. And then we need them to establish the right connections. The cells that we introduce need to extend these fibers that will connect the retina with the brain, as Dr. Welsby shown before. And so where do we get cells for these transplants? Luckily, we have technologies that we can grow retinal ganglion cells, and for that matter, any cell of the retina in Petri dishes in our lab. In 2006, Shinya Yamanaka won the Nobel Prize thanks to developing this technology called induced pluripotent stem cells. And what this technology allows us to do is to take any cells. In most cases, we can take a little bit of skin from a simple biopsy. We can grow skin cells, or called fibroblasts, in the lab, and we can reprogram them and change them so they become very similar to the cells that make up an embryo. And what's wonderful about the cells that make up an embryo is that these cells have the potential to make any organ in the body. Of course, during gestation, an egg starts to divide, and from one cell you get 2, then 4, then 8, etc., etc. But those cells are able to make all the body, including the eyes. And so now we can take any cell and make them very similar and have the same properties of these cells that make up the embryo, called embryonic stem cells. The ones that we make from skin or from any other source are called induced pluripotent stem cells. And not only we can grow these cells in the lab and we can culture them, we can also differentiate them by putting them in the right conditions. We can make them become retinal ganglion cells, or the cells that die in glaucoma. This is a protocol that takes many days, sometimes takes months, but at the end, we can get many of these cells. And not only the cells get the right identity and the right molecular signatures and many of the components of the cells, they also have the same morphologies, including the fact that they extend these fibers that will make the optic nerve. And so we can make these cells in a lab, and we got pretty good at this. But I don't want anybody to get confused. We are not growing eyes in the lab, but we are growing at these tiny, tiny pieces of tissue that can contain these cells. These organoids, these mini retinas, cannot grow too much. They are not vascularized. They don't have blood vessels. And because of that, what we can grow is limited in size, but we can grow many and many plates of these cells. So that is working really well. And as Dr. Welsby told you about, as a group, we've been working on several neuroprotective strategies. And he mentioned 2 new ones that we just developed, but in the past, we have identified other very interesting molecules, including the GCK4 family of proteins. That's also a very important target for neuroprotection strategies. And so not only these neuroprotective strategies work really well to protect the cells from the retina from death. They also work really well in the cells that we make in these dishes from these stem cells to get them to not only survive better and be healthier, but also they are able to extend longer axons when we treat these cells that we make in the lab with these molecules. And so we can make cells and we can get them to survive in a dish and also when we transplant them. So these strategies have really improved the survival of the cells after we transplant them in the eye. So we can follow the cells that we inject in the eye have been labeled with this green fluorescent protein, so we can follow them over time. And when we don't do anything, we just make the cells in the lab, we inject them in the eye, and we follow them over time. We see some cells surviving, but many of them over time die. When we treat them with these neuroprotective strategies, we have much better survival. So that's all very encouraging, but we also encounter a very important roadblock, and it's the fact that very often the cells that we inject in the eye do not really integrate and connect with the retina. Instead, they stay alive for many months, but they stay alive inside the vitreous, inside the jelly liquid inside the eye, but they don't really connect with the retina. And of course, having cells that are not connecting with the rest of the film of the camera and are not getting any information from the light is not really useful. So what can we do to really promote that the cells that we transplant really get in the retina tissue? So together with Dr. Welsby, we tried a surgical approach in which we can take these very, very small forceps and make very small openings in a membrane that separates the vitreous, so the inside of the eye, from the retina. This membrane is there to really protect the retina, but we can surgically open it up and ask what happens if this membrane is open, and then we do a transplant. Does that improve the integration of the cells transplanted with the retina tissue? And the answer is yes. This was an effort led by Dr. Derek Welsby. And so we see that if we don't do anything to the retina, we have some of these cells mostly staying outside the retina, staying on top of the retina, but not really integrating. And there's few of them and sort of sparse. When we open this membrane, now we have a lot more cells really in that spot. And we start to see that some of these cells can connect and send little projections and get information from the rest of the retina. So this has been really promising approach. But while doing all these experiments, I also started to really be concerned about another possible roadblock, and that was the immune system. So one consideration that we need to keep in mind is whether when we do these transplants and we inject cells into an eye, whether we are triggering an immune response that could really affect if the cells can survive or the cells that we put can connect with the rest of the retina. And the answer is yes. It's not the injection itself. We injected just nothing, just a little bit of liquid in the eye, or we injected the cells that we make. And we saw that when we inject just like a little bit of saline, a little bit of water in the eye, nothing happened. These cells here are the immune cells. They're normally surveying the environment and making sure that there's nothing going wrong, there's no infection, there's not an insult happening in the retina. So an injection itself doesn't do anything. But when we inject the retinal ganglion cells that we make in the lab, we start to see these cells reacting, getting really big. And these cells, these immune cells, have the ability to engulf and really eat other cells. So there's a real risk that if we trigger the immune system, when we inject new cells into the eye, these immune cells could be eating the cells that we put there, which would be obviously not very good. And so that raises another concern, and is that, of course, we will never, in a clinical setting, use transplantation in a healthy person, in a person that doesn't have glaucoma or doesn't have a retinal ganglion cell degeneration going on. And so instead, if we do transplants in the clinic, will be in the context of an ongoing degeneration. And so now this is the experiments we are doing right now in the lab. We have models of degeneration. Dr. Welsby just showed you one, which is this model where the mitochondria doesn't get transported really well. But we have other models that mostly also affect mitochondria and lead to this degeneration of the retinal ganglion cells, this loss of axons. And now we're asking what happens in these models when there is an ongoing degeneration and we do the injection. And what we know that happens is that the ongoing degeneration also activates the immune system. And so these cells, these immune cells, are already reactive, are already noticing that something is happening in the retina, there's inflammation, and they're more prone to be eating other cells. They eat the retinal ganglion cells that are dying, so they're already in this form where they are prone to eating other cells and reacting to other cells. So a big concern is that if we inject cells in this situation where the immune system is already active, it will be a lot harder to have the cells to survive. So what we are doing and what we're going to be working in the next few months is try to modulate the environment and try to modulate this immune system. And we have developed a lot of tricks to really silence this immune system so we can inject the cells and ask what happens when the cells are not injected, both in control situations in a healthy eye, but also in a situation where there's an ongoing degeneration and there's retinal ganglion cells dying. So, of course, the current state of the replacement strategies is not as advanced as the neuroprotection advances, but I'm very excited because in the last few years, we made so much progress. We learned how to grow very effectively retinal ganglion cells in a dish. We know how to get the cells to stay very healthy. We have developed neuroprotection strategies that can not only protect the cells while they're in the dish, in the lab, but also while we transplant them. And we can get these cells after transplantation to survive for months. We had some for 13 months in a host eye. So that's all very promising. So I'm hoping that really the manipulation of the environment would be one really good trick to get the cells to survive better and to really be able to integrate in the context of an ongoing degeneration. And so with that, I just want to leave some time for questions, but I cannot finish without really thanking the Glaucoma Research Foundation for all their support and all the members of the CFC team, including Dr. Derek Welsby, Xin Duan, and Yang Hu. Thank you very much.
Well, thank you, Anna and Derek. Those were just fascinating presentations, and I'm sure we will have a lot of questions, and we have some that have already been submitted. And of course, the first one I have to ask, which is one of my favorites, is how quickly do you think that we will see some of that research actually being available to patients? And so I'm going to ask you, Anna, maybe first, when do you think you might see actually restoration of vision with implanting new cells? And then we'll ask Derek what his projection is for neuroprotection, where we're protecting the existing retinal ganglion cells. So maybe you can each comment on this question, please.
So, of course, Derek will be more optimistic about the neuroprotection, I suspect. But for cell transplantation, we made lots of progress. So I'm really optimistic, but there's a lot of challenges that we still need to overcome. We got really good at cell production, at survival, and now comes the hard part, which is getting the cells to really connect. And the connection has to be two ways. The cells need to connect with the rest of the circuitry of the retina and receive all the information from the light, and then send all that information to the right parts of the brain. We've been tackling challenges as they come, one step at a time, but I'll suspect this will take a few years until this is ready for the clinic. That being said, technology moves at really exponential speed, but it will take a few years until this is ready for a clinical trial.
Derek, maybe you might have a little more interesting news for our participants.
Yeah, I would say, look, neuroprotection is happening now, and it's coming in many different forms. For instance, there are clinical trials right now to evaluate vitamin B3. Frankly, people can take vitamin B3 right now. I think there's still equipoise about whether that's the right thing to do, whether it could make the disease worse, better. So fortunately, there are clinical trials to evaluate that, but that's, present time. Then you have clinical trials for things like insulin eye drops, where those are already in humans for clinical trials. And then you have some of the neuroprotective therapies that you've heard about through the GRF and the Catalyst for the Cure. And those are starting human clinical trials, or they're being developed for non-human primates. So they're not quite as far along. But I guess the point is, it's really starting now, Tom. And over the next 5 years, I think we're going to see this wave of things being evaluated so that hopefully, let's say, 5 years from now, which is really not that far away, that there really will be different options for patients in the clinic.
Yeah, that is very exciting news. Another question was, how are they able to access these clinical trials? And I wonder if maybe you could comment on that, because I know patient recruitment is always an issue, but which patients would be, let's say, qualified for a neuroprotective clinical trial? How are they selected? And if someone would like to be part of a clinical trial, how do they get access?
Right. Anna, can I take this one?
Yeah, of course.
So I would say, first off, there is a list of clinical trials on clinicaltrials.gov. So if patients want, they can go there. But it's a little bit of a dense read. It's not the easiest thing to interact with. Honestly, I think the Glaucoma Research Foundation as an educational site is probably one of the top places that patients should go to figure out sort of what's exciting. And then I would say, frankly, it's your glaucoma specialist. And especially glaucoma specialists who are plugged into university settings, although it doesn't have to be that way, are really going to know what clinical trials are out there, which ones might be appropriate. Your question about who's going to get selected for what clinical trial, it really depends on what the therapy is. It depends on the risk-benefit profile of that therapy. And it depends how early the trial is. So you could imagine that when you're first testing a new agent, you don't want, there's really the number one thing is do no harm. So you want to make sure that you're testing this in relatively severe disease. So God forbid, if there was a problem that, you haven't blinded, for instance, a perfectly functional eye. But then as you are trying to demonstrate that your therapy actually works, you can't use an eye where it's already lost all of its optic nerve cells because there's no way to show it that your therapy would work. So, it really depends on what stage of the clinical trial that product is at.
Yeah. So I would encourage our participants to look at clinicaltrials.gov, because you can search it. So you could put in glaucoma and neuroprotection, and it will show you the active trials and where they are And then you could even take that list to your ophthalmologist or your glaucoma specialist, and maybe they'll know someone and be able to connect you. But as Derek said, it really depends what stage of your glaucoma journey you're on and where the trials are and so forth. So another question is, what role do you think AI? So everybody talks about artificial intelligence, and I don't think anybody really knows what it is, but it certainly is a powerful tool to analyze data, and it is being used by the Catalyst for a Cure. So Derek, where do you see the role for artificial intelligence in your developments?
Oh, I think it's going to be pervasive throughout all of medicine. And if I had to pick the areas that are most relevant to this talk, I would say, look, it's understanding what's the trajectory of a given patient and using genetic data, demographic data, history about their imaging, what's happened to them so far, their treatments. Projecting the course of a given patient helps both the care of that patient, but also it helps in the design of the clinical trials, where you can really cherry pick those patients who are the ideal candidate for a given therapy. Also, we might be able to find, when we do clinical trials, often we just take a bunch of glaucoma patients, you randomize them to, treatment, no treatment. And it may be that the treatment isn't a one-size-fits-all, that there are certain patients that benefit more from a given treatment, what we call sort of personalized medicine. And I think finding that pattern is not always so obvious. It's not just one thing. So AI is very good at pattern recognition and understanding these patterns and making those predictions. But I mean, AI is going to help us understand the literature. It's going to, it's just going to change all of medicine.
It is sort of amazing. Are you using it now in some of your work?
A little. So for some of the experiments we do right now, because technology is advanced so much, we have data sets that are enormous. And it's really difficult to sort through that amount of data and to really find, patterns and differences. And so we do use a lot of machine learning, and, machines can do the sorting for us for many of the experiments that we are using right now, and to look at big data sets. So yes.
So another question for both of you, and I think each of you answered this a little bit, but is the optic nerve damage reversible?
The optic nerve damage is reversible. So so we kind of talk about neuroprotection and preserving the cells while they're still there. And we also talk about cell replacement. And we kind of skip through something in the middle, which is called regeneration. It's like there's strategies that are being trialed that when a cell is still alive, still there, but has lost the fiber, the connection between the retina and the brain, there are strategies to regrow that axon, that fiber that's part of the optic nerve. And so there's something in between neuroprotection and cell replacement. Of course, preserving the optic nerve when the cell is not there, or improving the optic nerve health when the cell is no longer there, is not an option. But there's room for growing these fibers again. I don't know if that.
To add to what Anna said, I think there's another middle ground, and that is this idea that as the disease is happening, there may be a population of optic nerve cells that are damaged, not functioning particularly well, and that by using various neuroprotective strategies, pressure lowering, whatever the therapy might be, that you may sort of wake them up and allow them to contribute more to vision. We don't really know how big that population is. Personally, I don't suspect it is a large effect. There are clinical trials right now using things like electrical stimulation that attempt to either, wake up that population, or perhaps more appropriately described, they're trying to get neighboring cells to take up some of the slack of the cells that are injured or dead. And again, these things would give a patient some reversibility, but it's not going to be a huge effect where you're going from big blind spot to seeing in that area. That's going to require Anna's magic.
So that kind of raises another interesting question, which is, what degree of blindness and the length of time vision has been lost? How does that affect the possibility of either rescue or restoration? And you both sort of talked about that, but if someone's had glaucoma for a very long time and lost a significant amount of vision, is that a more difficult case, whereas someone who's early on and just beginning to lose vision might be a more somehow respond more to some of these new treatments? What's your thinking there?
I would just say, I think it's a great question. And frankly, Tom, it's an area that is actively being investigated and actually probably needs more attention. And I'll tell you specifically, it has to do with the brain centers. So when you have areas of the brain that haven't been connected to an eye for long periods of time, we know there's atrophy. That's been seen on MRI. And the question is, okay, someday we come up with a perfect therapy to replace those optic nerve cells, get them to regrow to the brain. You can do the greatest job possible on those optic nerve cells. We don't really know what is the capacity of those brain centers to start to take that input again and make functional vision. I think there is evidence that it could happen, but I certainly think this is an area that is just sort of an open question.
Does your research apply to people of all different ages? So we tend to think of glaucoma as an age-related disease, but in fact, there are children and young adults who also have glaucoma. Do you think the things you're looking at could be applicable across the age spectrum, or would be, what's your thought on that?
Yes, I don't think the age is going to be a limit for any of the strategies we are using. In fact, which is sort of ironic, for the transplantation, we know that sometimes when we inject the cells, the cells that we inject get stuck in the vitreous. This is like the jello-like substance inside the eye. And as we all age, that substance it becomes a little more liquid. So hopefully that actually makes it better. And we have some evidence that that's the case. But generally speaking, there's no age limitation for any of these therapies.
Yeah, we tend to think that there probably are some common pathways, common mechanisms of how glaucoma damages the optic nerve. And, obviously there's a lot of interest on the typical glaucoma that happens in older people, some of which have high eye pressure, some of which don't. But funny enough, all of our animal models always use elevated eye pressure. Actually, the one I presented today, Young Hu's model, is one of the very few that doesn't use elevated eye pressure. But we know a lot about how to treat elevated eye pressure in animals. And I will just say that when you talk about glaucoma in humans, while people with older glaucoma, it can be high eye pressure or not, if you're talking about younger glaucoma, like really under age 30, those are almost always cases with elevated eye pressure, which really does match the animal models that we're using in the lab even better. So I suspect that probably things will translate across ages. Not to say that age won't be an important variable that we'll need to understand better.
But that's encouraging to know that there, could be techniques or therapies at all different ages and maybe at all different stages of the disease. And you're looking at all of that. And again, that's a great place where AI can help, because you can look at larger populations of patients and larger symptom bases and try to tweeze out the, let's say, the right parameters for a particular therapy. So that's pretty exciting. Another question that I have is shifting a little bit from what you're doing right now, but speaking more generally to the question of how do patients, are there things they can do today, you mentioned vitamin B, for example, but are there things that people can do today that might help to prevent their glaucoma from getting worse? Of course, I know one of the answers right away, which is work with your doctor and follow the directions and take your medications, the eye drops, and all the rest of it. So that's probably number one. But I'm sorry, Derek, I'm asking you the question, and I'm not going to answer it.
I would just say the vitamin B3, that was not a recommendation because I think there is evidence that there are reasons to believe it could even be harmful. And so whether it be side effects of the medication or even the pathophysiology of glaucoma. So I think we need clinical trials to understand whether that should be a recommendation. Just want to clarify that. I think there is pretty good evidence for two things. And it's not the best evidence, but cardiovascular exercise and green leafy vegetables. I feel pretty comfortable, even without the best of evidence, recommending that to everyone. Eat healthy, exercise. There is some evidence that can help glaucoma, in addition to what you said, which is just engage with your doctor and follow the plan. And the reality is most patients in that situation do pretty well.
Yeah.
So it works pretty well. It's just not perfect. And that's the reason we want to have these sort of innovative approaches.
Well, along that line, I think also for participants to remember is to make sure their family members know they have glaucoma. And that comes back to getting diagnosed early and treated early, and that helps to preserve vision. And we do know that there can be a genetic aspect to glaucoma. So making sure family members know and taking the opportunity to be sure that your family members are screened for glaucoma is very important as well. Can you think of anything you would say, things to be avoided that come to mind to prevent glaucoma or increased eye pressure? I think that's a tough question. I don't know.
I guess choose your family members carefully.
Well, I know there are some exercises that are not used, but that's a rare situation, I guess.
Yeah, so there are situations of people who play certain musical instruments, do certain types of yoga positions, but no general recommendation for a population. That's a conversation between you and your glaucoma specialist. Maybe in very, very select circumstances, you might want to avoid something, but generally speaking, no.
So you mentioned clinical trials. Are there nutritional trials that you're aware of? And I guess you could look, some of these may not, if they're supplements or whatever, they may not be on clinicaltrial.gov, but I don't know if there are any trials that you mentioned. The electrical stimulation trial, which probably is on clinicaltrial.gov and has been done. I think it's being done at Wilmer and Stanford and Columbia, I believe. But are there any that come to mind of a nutritional
or sort of like nutraceutical world? Not that, besides like vitamin B3 and pyruvate, not that I know of. It's often when you see evidence in the literature or studies that support use of some sort of nutraceutical, it's what we call a retrospective study, where you've looked at people and you sort of divide up the past into people who use something and people who didn't happen to use something and see if one group did better. There aren't too many, the better type of evidence, which is what we call a randomized clinical trial, where people upfront are told you're in this group or that group, you're going to be given that or that. We follow you over time. That's much less common for the nutraceutical studies. There's none that I'm aware of besides the ones that I've mentioned. But again, I'm not an expert.
I wonder, Anna, can you comment on, I'm just thinking on the cell replacement and vision restoration front, what kind of work is going on around the world in that? I mean, I know you're on the kind of leading edge, but I think that you came back from a meeting in Spain, I believe, not too long ago. So can you give us a little bit of an idea of how active that field is around the whole world?
It's very active. So people have tested several ways to inject cells. I think the experiments that we did over the last maybe 3 years, we all came up with the same conclusion that that membrane, it's called the inner limiting membrane of the eye, was really a barrier. And so we have been exploring surgical methods to open up. Other groups have been testing enzymatic methods to open it up. And other groups have been testing whether injecting in the opposite side of the retina from the outside of the eye is actually better. The injecting in the other side of the eye gave mixed results. When you inject cells in the other side of the eye, the cells tend to stay there. And so those need to be supplemented with a molecule in the eye to guide the cells to move. And the success has been moderate. So there's some interesting developments, but the success so far has been moderate. And I think the best results have been injecting directly when you want the cells. And people have also explored, different ways to manipulate the environment to make these transplants better, and using different models to do these transplants. But there's a very active research going on throughout the world. So stay tuned.
I mean, it has to be very encouraging for glaucoma patients that they're, it's not just Catalyst for a Cure, but it's many groups around the world, and they talk with each other and share ideas. And
Absolutely.
so it's good to know that a lot of people are working on these problems. So I want to thank you both for making time to be with us today and for your dedication to advancing glaucoma research and helping glaucoma patients to preserve their vision. And thank you to our participants for your interest and support. If we were unable to answer your question today, please visit our website, www.glaucoma.org, for the latest information about glaucoma and our research. The important work done by Glaucoma Research Foundation and the progress towards a cure is made possible by generous supporters like you. You can help us to speed the cure by making a one-time or a monthly donation or including Glaucoma Research Foundation in your estate plan. While continued financial support is critical to our path toward a cure, there are additional ways you can help. Be our champion and advocate. Share our story with family and friends. Let your doctors know about Glaucoma Research Foundation and share with them our excellent patient resources. And lastly, share with friends and family the importance of regular eye exams. Everyone is at risk for glaucoma, and early detection and treatment are essential to preserving sight. Thank you once again for being with us today and for joining us in our bold vision of a future free from glaucoma. Because of your continued partnership, the cure is truly in sight. See you next time.